Thank you for joining us today for our webinar, Preventing Anaphylaxis in Those with Food Allergy. Is it possible? I am Jennifer Gertz, Executive Director of Food Allergy Canada, and our guest speaker today is Dr. Manel Jordana. Just a couple of housekeeping notes before we get started. Note that the session is for informational purposes only and will not provide specific medical advice, recommendations, diagnosis, or treatment. All participants are muted so we can keep the audio clear for the webinar. We did receive a number of questions on registration, and we will accept some questions during the session through the chat box throughout the webinar. At the end of the presentation, we'll try to get to as many of those questions as we can with the time we have available. This webinar will be recorded and shared on foodallergycanada.ca afterwards. Before introducing our expert, I'd like to frame up the content for today's session. This session really covers the science behind an allergic response and anaphylaxis, the mechanisms, what's actually happening in our immune systems, and the implications for preventing anaphylaxis in future treatment. So it's helpful to understand how this then fits into the development of treatments and therapies. So what you see on the screen in front of you is the typical process that's used by researchers and pharmaceutical companies to understand disease states, understand underlying mechanisms, and to develop therapies ultimately to help patients impacted. So it starts with basic science, the in vivo, the in vitro within the lab. It then moves on to clinical trials and clinical developments to ensure both its safety and the effectiveness in humans. And then that review of clinical data is taken a look at by regulators. So in the case of Canada, it's Health Canada that would look at that data and would make its decision on whether they would approve a therapy. Then work is done on how the therapy will be funded, and it's rolled out to market, and data is continually gathered to continue to advance the therapies that have been approved. Today, we are so fortunate to have one of the experts and frankly pioneers in the field of food allergy, whose focus has been on really the first part of the therapy development cycle. So the area in pink and purple there. Since 1988, Dr. Jordana and his research team has had a focus on food allergy and has made significant contributions to the knowledge base, essentially starting with what might be considered a blank page to now informing the world of researchers globally with insights on the mechanisms underlying food allergy and the possibilities for preventing anaphylaxis. Several of you in your questions submitted on registration asked about whether the learnings presented here are applicable to other food allergens beyond peanut. And so we don't want you to have to wait till the end of the session to have that question answered. The short answer is yes, but we will discuss that again at the end of the presentation. Now I'd like to introduce our guest speaker. Dr. Manel Jordana is an MD, PhD, and professor in the Department of Medicine at McMaster University. He joined the McMaster faculty in 1988 and has since supervised over 75 trainings and co-authored 189 scientific papers, reviews, and book chapters. Dr. Jordana is the lead investigator for the new research being shared today, which is also featured as an editor's choice in the April issue of the Journal of Allergy and Clinical Immunology, a strong indication of the importance of this work. We are delighted to have this expert spend the next hour with us in helping us understand the science of food allergy and this question of preventing anaphylaxis. Is it possible? Dr. Jordana, over to you.
Thank you, Jennifer, for this introduction. Good morning, good afternoon to everybody. A pleasure to be here. And I have to say that I was really delighted to accept the invitation from Food Allergy Canada and the title that they proposed. The invitation from Food Allergy Canada came as a result of a paper that has been published just a few weeks ago in the Journal of Allergy and Clinical Immunology. This is the paper. Now, in the agenda for today is as follows. First of all, we'll talk about some basic concepts. Then we'll talk about the process that led us here to this particular research. And I am particularly interested in sharing with you the narrative, or if you wish, the logic of our research project. Then I will present to you some key findings from this study and the implications of these findings. Now, typically, one gives a talk, and then at the end, thanks people and organizations, but sometimes one forgets because of time or any other reason. So I'm going to switch things around and acknowledge and thank right off the bat all of the people who contributed to this work. This is the important people. This is the core group of graduate students who actually did the work. A group of very young, as you can see, and very talented students. And specifically, I want to acknowledge Kelly Bruton and Paul Spiel, who led that project. The other important people is all of those organizations who so generously funded our research. So let us begin. First of all, some basics about antibodies. Antibodies, or immunoglobulins, are a group of molecules that are truly essential defense mechanisms. There are different classes of immunoglobulins, also referred to isotypes, and they have different functions and mechanisms of action. For example, in the case of IgG, it plays a very important role to fight viral infections and is the type of antibodies that are generated with vaccinations. Then IgE also is a very important, participates in a very important defense mechanism against parasites, venoms, and toxins. But of course, the flip side of this is that IgE is the primary immunoglobulin mediating allergy. And in the case of food allergy, is the principal molecule that mediates allergic reactions. Now, what are the key players in an allergy reaction? First of all, as I mentioned, IgE. But the other key player or players are what we refer to as the allergic cells, mast cells and basophils. Mast cells are distributed throughout the body in mucosal surfaces and the skin. We all have these cells all the time. And basophils are not tissue-resident cells, but are circulating cells. Now, note this picture. This is an electron microscopy picture of a mast cell. And you can see in the middle, there is the nucleus. And then within the cytoplasm, there are all of these dense structures that are called granules. And these granules contain a variety of preformed mediators. Now, this is another representation of a mast cell containing all of these granules. In blue, you can see IgE, which is bound to the mast cell or to the eosinophil, to the basophil, through a specific receptor. Now, right on top, you can see the allergen. When an allergen, in an allergic person, when an allergen comes along and binds, cross-links to IgE molecules, there is a process that it's called degranulation. And that means that all of the products contained within mast cells or basophils are released into the microenvironment. This is a list, not exhaustive, but it's a very complete list of the kind of molecules that are released during degranulation. Histamine, heparin, serotonin are molecules that are contained in those preformed granules I was referring to. And then other molecules such as PAF, platelet activating factor, leukotrienes, prostaglandins, proteases are released, are synthesized very quickly and released within minutes. Now, these are precisely all of the molecules that contribute to these clinical symptoms: sneezing, coughing, tearing, vomiting, swelling, diarrhea, itching, and then, of course, in extreme cases, anaphylaxis. However, if we now go back to the flip side of an allergic reaction, if you think about all of these symptoms also serve another purpose, and that is the expulsion or containment of parasites, venoms, or toxins. So what is good for one defense process is harmful for another, which is the response to allergens, specifically to food allergens. Now, given the critical importance of IgE in food allergy, then we started asking questions. For example, who makes IgE? IgE is made by cells that are called plasma cells. And this is a schematic of a plasma cell. And I want you to notice right in the middle, this purple nucleus. And then all around, there are these structures in green color. This is something that is called rough endoplasmic reticulum. And this is literally a huge factory that produces immunoglobulins at an outstanding rate. All kinds of immunoglobulins, not only IgE. Indeed, they produce 20,000 molecules of immunoglobulins per second. So, of course, the next question is, how are IgE producing plasma cells are made? But we need to go back and ask a more general question, which is, where are immune responses generated? Well, this is the lymphatic system. And the lymphatic system is a system that runs in parallel to the circulatory system. Imagine for a moment that you have a big highway that you call the circulatory system, the circulation. Well, then all the service roads that run in parallel to this highway would be the lymphatic system. At a closer view, throughout the lymphatic system, there are these structures in red that are the lymph nodes. They are distributed throughout the body. And then different parts of the body drain to different nodes. Many of you may have noted that, if you have a severe throat infection, some of the lymph nodes in your neck become enlarged. And this indicates simply that there is an immune response that is generated. So let's now take a look at one of these lymph nodes. Now, this is an image, a cross-section of a real lymph node. Lymph nodes are fascinating structures. With this technology, one is able to see that every color that you see represents a different cell type. And you can see that these different cells are distributed differently throughout the lymph node. So the generation of an immune response, any kind of immune response, involves a very complex crosstalk among all of these different cell types. And it's a crosstalk that involves many different molecules. Now, let's now simplify the lymph node using this schematic. And let us also focus on one specific immune process, which is the generation of allergic humoral immunity. In other words, the generation of allergic antibodies. Here in these lymph nodes, I remove all the names because they're not really relevant at this point in time. But essentially, what happens is that allergen, which is transported from the periphery into the lymph node, engages in a series of complex interactions with all of these cells. The outcome of this crosstalk is the generation of two different types of cells. One, long-lived IgE plasma cells. In fact, what exits the lymph node is a precursor that is called a plasmablast that migrates into the bone marrow and sits there and start producing immunoglobulins. In this particular case, IgE. This IgE would be dumped into the serum and then become cell-bound to either mast cells or basophils. But then there is another cell type that is generated, which is a memory IgE positive cell. And these cells just sit there. They don't proliferate. They don't do anything, but they are able to remember the antigen, the specificity that generated these cells. So when this allergen comes along, they are able to become activated. Now, the most important part of this slide is what you see right at the top in yellow. This is a hypothesis. And in fact, this hypothesis was generated as an extrapolation of what we know that it happens in the case of IgG, the type of immunoglobulin that is crucial in vaccinations and in the fight against infections. And it was assumed that if this is what happens with IgG, the same is going to happen with IgE. But that remained unproven. So this is the end of basics. With this knowledge now, we can ask some other specific questions. Let us begin here. A critically important clinical observation is that some food allergies, such as those to peanuts, tree nuts, shellfish, are lifelong in the majority of patients, about 80% of the patients. Now, intuitively, one would think that this is because IgE is constantly produced. And that would imply that there are long-lived plasma cells that make this IgE. And certainly that would be consistent with what we know about IgG and viral infections or vaccinations. However, the levels of allergen-specific IgE fluctuate in patients with seasonal allergies. They are more elevated during the season, and then they decrease during the off-season. Allergen-specific IgE levels plummet in patients allergic to an anisakis, which is a parasite in fish. But these levels decrease dramatically when the person successfully avoids fish. Excuse me. And finally, we also know that these levels of allergen-specific IgE decline over time in patients that are allergic to something that is called alpha-gal when this person avoids tick. It's a fascinating story, but it's beyond the scope of this presentation at the moment. But you see, the fact that you see all of these fluctuations in the levels of allergen-specific IgE indicates that these are not consistent with the existence of long-lived IgE-producing cells. So here we face the first conundrum. If IgE-producing plasma cells are not lifelong, what makes peanut allergy persistent? So we need to go back to 2013 here. And at this point in time, we initiated a project to investigate various factors, most of the factors that could contribute to the persistence of peanut allergy. Now, the result of this inquiry was this paper that was published in 2017. And the title, I think, that it's explicit enough. We found that the persistence of peanut allergy was due to the existence of lifelong memory cells, not plasma cells. I'm going to show you some of the data in this paper to convince you that this is the case. Now, for practical and ethical reasons, it is imperative sometimes to use animal models, because this is the only system where we can answer certain questions. And of course, these models provide an unparalleled spatial and temporal control about all the variables that are in place. Effectively, we can make mice allergic to any protein that we want. And we can use different models. And today you will see two. The first one is a model of oral sensitization. So what we do is we administer into the stomach peanut along with an adjuvant that is called toxin on 4 occasions, 1 week apart. And this initial phase is called the sensitization phase. And the purpose is to generate peanut-specific IgE. At that point, we wait. And we can wait 1 week, or we can wait up to 15 months doing nothing else. And then we challenge the mice. And what we can do is we can evaluate whether or not there is anaphylaxis by measuring changes in core temperature, hematocrit, blood pressure, and we can measure all sorts of molecules in different compartments. This is essentially what we measure to evaluate anaphylaxis. Right at the top on the left, there is temperature. The y-axis indicates degrees of temperature, and the x-axis indicates time. And I want you to notice it's time in minutes. And what you can see is that right after challenge of a sensitized mouse, there is a very rapid and robust decline in temperature. Now, this experimental is very important. In humans, whether the ambient temperature is +30 or it's -30, our core temperature is always 37 degrees. And the reason is that we have very robust thermoregulatory mechanisms. But these mechanisms are non-existent in mice, or they are very poor. So when mice become ill, the temperature drops, and the more severe the illness is, the greater is the temperature drop. Along with the decrease in temperature, as you can see on the right, there is also a very significant decrease in blood pressure. Bottom left, there is another measurement that we perform, which is hemoconcentration. In the course of an anaphylactic reaction, there is vascular leakage, and that means that plasma leaves the circulation. So the cells that remain behind, especially red blood cells, become thickened, and this is what it's called hematocrit. So in the course of an anaphylactic reaction, there is a very substantial increase in hematocrit. And lastly, we also evaluate a clinical score, which goes from nothing, 0, to 5, which is tremor, convulsions, and death. But we are not allowed to get to number 5. So once mice reach 4, then we initiate the rescue process. So let's go back now to the data. This is the first piece of data that I wanted to show you, which is levels of peanut-specific IgE. In the y-axis, you can see values that indicate the amount of IgE present in the circulation. And then on the y-axis, you can see time in months. So you can see that experiment requires a lot of patience from our part, because in some of these experiments, we waited 15 months. Take into account that the lifespan of a mouse, it's about 2 years, more or less. You can see that there is a dramatic decrease over time in peanut-specific IgE. So what that shows is that in the absence of re-exposures, the levels of peanut IgE are not persistent. What is the clinical significance of this change? Well, here you can see on the left is a temperature change, and on the right is hemo concentration or hematocrit. The temperature change here is represented a little bit different, and this is the reason. You can see in the y-axis, in the x-axis, that it's time in months: 1, 3, 6, 12, and 15. So each time point was examined in one separate group of mice. So in other words, mice were never challenged twice. Some mice were challenged only at 15 months, others at 12, others at 6, etc., etc. So if we try to plot temperature in the way that I shown to you before, it would be a mess. It would be so many lines that it would be actually quite messy. So what we did here is to calculate the area under the curve of each one of the curves. So what you can see here is that, for example, at 1, the temperature change is very great, but as time goes on, it decreases. And likewise, when you look at hemo concentration, you can see that there is a progressive decrease over time in the thickening of the blood. So in other words, mice no longer react to challenge if they are not re-exposed. This is the next experiment that we did. We sensitized mice. We waited either 9 or 15 months, and then we re-exposed the mice to peanut without any adjuvant. Sometime later, we challenged the mice and we evaluated for the presence of anaphylaxis and other things. Now, the first one of these graphs is, again, antigen or allergen specific IgE. And I want you to notice the x-axis. As expected, at 9 months, there is no IgE in serum. But when those mice were exposed to peanut, there is a sudden increase in IgE levels. The graph on the right shows an additional piece of direct information, and that is we evaluated the number of allergen-specific IgE-producing plasma cells in the bone marrow. And again, as expected, at 9 months, there are no IgE-positive plasma cells. But upon re-exposure, then there is a sudden increase, consistent with the increase in IgE in the serum in the bone marrow. Again, what's the functional significance of these changes? So in terms of hypothermia and in terms of hemoconcentration, in this particular case, the evaluation was performed at 15 months. And you can see that the 2 lines at the top, one of them is naive, normal mice, non-allergic mice, and the one with the closed squares is mice that were sensitized and then challenged at 15 months. You can see that there is a flat line. So in other words, the mice did not react, did not go into anaphylaxis. However, in those groups of mice that were exposed to peanut, either systemically or orally, they developed a very acute anaphylactic response. And you can see the same thing on the graph on the right, where we looked at hemo concentration. So with that said, that even though IgE positive plasma cells are not lifelong, the capacity to regenerate IgE is lifelong, and consequently, so is clinical reactivity. Now, let us consider, the initial working hypothesis. Basically, what these studies show is that IgE producing plasma cells are not lifelong. At this point, we came up with another, hypothesis, and we said, well, maybe the cell type that is lifelong is the memory cell, and specifically a memory IgE positive B cell. And the argument was that in the context of allergen re-exposure to someone, a person or a mouse that is allergic to peanut, allergen re-exposure will activate these memory IgE positive B cells that will differentiate and become IgE producing plasma cells. That was our next hypothesis. The problem was that another project that was ongoing in the lab at the same time was investigating the existence of alleged putative IgE positive memory B cells in peanut allergic persons. And the result of this experiment was this paper that was published in 2019 and effectively demonstrated that IgE positive memory B cells in humans are either extremely rare or they don't exist at all. So here was our next hypothesis, basically down the drain. And that presented a second conundrum. If IgE positive memory B cells do not exist, what facilitates the virtually lifelong capacity to regenerate IgE? Now, I'm now going to show you what it's the current understanding about the path that regenerates IgE. Fortunately, we have learned more about the origin and the regeneration of IgE in the last 5 to 7 years than in the past 100 years. And I think that this basic understanding has become really fundamental. The current understanding is that there exists a lifelong memory B cell, but this cell is a non-IgE memory B cell. Remember that our previous work showed that the IgE memory B cells basically don't exist. So what kind of a cell is this one? Well, in the mouse is an IgG1 memory B cell. And we know that in humans, there are several isotypes, but primarily is IgG1 and IgA1. So in other words, as you can see that there are cells, memory B cells, that are lifelong and of a different isotype than IgE. So what happens is that when a mouse, or presumably a human who is allergic, is re-exposed to this allergen, again, there is a crosstalk that takes place between this non-IgE memory B cell and another cell type that it's called a CD4 positive T cell. As a result of this crosstalk that involves certain types of molecules, this non-IgE memory B cell becomes, through a very complex process called class switch recombination, an IgE positive B cell. This cell proliferates and it terminally differentiates into an IgE producing plasma cell. And this is the cell, of course, that produces IgE. So this is the most updated and current understanding about how IgE is regenerated in allergic persons. Now, this schematic here is essentially the same as the one before. You can see on the left that there is that T cell, a CD4 positive T cell. On the right, in green, there is the IgG1 memory B cell. And you can see here, as an additional information, that this T cell produces molecules that are called interleukin-4 and interleukin-13. These molecules act on a receptor called IL-4 receptor alpha on the IgG1 memory B cell. These are the molecules that facilitate the crosstalk and eventually the generation of IgE positive plasma cells. So we argued that if we use an antibody that blocks that receptor, so in other words, that it prevents the crosstalk between the T cell and the memory B cell, we will be able to prevent the generation of IgE positive plasma cells and therefore the production of IgE. And that was the concept, that was the hypothesis of the paper that was published just a few weeks ago. Here again, you can see Paul Spill and Kelly Bruton, the two ultra-talented students who led this project. Now, the way that we designed the testing of these hypotheses involved two different types of experiments, in vivo and in vitro experiments. So let us begin with the in vitro experiments. This is the schematic of the process. And basically, what we did is we established a novel in vitro platform using peripheral blood from either peanut allergic patients or non-allergic patients. So we obtained the blood, we performed something, a gradient, in order to obtain something that is called peripheral blood mononuclear cells. We place these cells in culture, we stimulate them with peanut, we wait a number of days, and then we evaluate IgE. Because IgE was the principal readout of these experiments, we wanted to be absolutely sure. So we used three different methods to evaluate IgE. One method is called ELISpot and tells us about the presence of IgE producing plasma cells. The second method is called RT-PCR and tells us about IgE mRNA transcripts. And the third method is ELISA, and that tells us about the actual amounts of IgE in the supernatant of these cultures. Now, and these are the results. And, you know, I just want you to focus on the columns with the red triangles. And basically what you see here is that with any one of these three methods, IgE secreting cells, levels of peanut-specific IgE, or IgE transcripts, peanut allergic subjects or cells from peanut allergic subjects that were stimulated with peanut generated IgE. Now you can see in the blue here, right at the bottom, all of these points, each one of these points indicates an IgE producing plasma cell. So that was a very robust system to evaluate the production, the generation of IgE. So then the next question was, so what happens if we treat these cultures with an anti-IL-4 receptor alpha antibody? And these are the results. And again, I just want you to focus on the red triangles now. And you can see that looking at any one of these three methods, the impact of the anti-IL-4 receptor alpha treatment was dramatic. So basically, it fully prevented the formation of IgE. Now, of course, the next question was, okay, so that's great. This is what happens in a culture system. But what happens in vivo? And for the in vivo investigation, we developed a novel experimental model. In this particular case, we sensitize the mice through the skin. And then under these conditions, the mice produced first peanut-specific IgG1. Then we re-exposed, no adjuvants of any kind, to peanut. And those re-exposures lead to the generation of peanut-specific IgE. And subsequently, any time after that, we challenged the mice. So as you can see, this was a very interesting system because it allowed us control of the transition from IgG1 to IgE. Essentially, and in some instances, of course, during the re-exposures, we administered an antibody treatment. Essentially, this is how it works. This is how we do it. So we take these mice and we shave the backs, and then we tape strip very gently the area that has been shaved. So this is very gently, and in fact, the tape stripping, the only thing that it does is to remove the outer layer of skin cells, which are dead cells. So in a way, it sort of generates a little bit of an eczema on the back of these mice. And in these areas, we then apply, we do not inject, we simply apply on top peanut. Now, these are the results. And this is a little bit complex, but we'll go through in an easy way. So whenever we do these experiments, we need to have lots of control groups so that we can interpret adequately the results. But here and now, I just want you to focus on the blue bar and the red bar. The blue bar are mice that went through this entire process of sensitization and challenge, untreated, no treatment. And the red bar indicates those groups of mice that went through that process, but were treated with the antibody during the re-exposures. And you can see in terms of serum levels of peanut-specific IgE, the difference between the blue bar and the red bar is absolutely dramatic. And again, we ask the question, well, what's the clinical significance of this? Again, focus on the red line versus the blue line. The blue line is right at the bottom. Significant, dramatic decrease in temperature, whereas the red line at the top, you can see that there is no change at all whatsoever. So these mice did not react in any way, shape, or form. And on the last panel, humoral concentration, if you again compare the blue squares with the red triangles, you can see exactly the same thing. And the red triangles are, identical to a naive mouse, so a mouse that is perfectly normal. One last piece of information. We did another experiment, and I need to explain the rationale for this experiment. Whenever you administer a treatment, whether it's an antibody or it's a drug, they all have a certain half-life. So they remain in circulation for a certain amount of time. So in the case of a drug, it may be hours, which is why sometimes you need to take your antibiotics every 12 hours or every 8 hours. In the case of an antibody, the time is longer. So the half-life of an antibody may be 1 week. What that means is that after 1 week in the circulation, there is only half the amount of the antibody that you administer. And after a second week, there is only a quarter, etc., etc., etc. So what we did in this experiment is we repeated, exactly the same thing, but we waited 6 weeks. We waited 6 weeks because at this point in time, we had done other experiments that ascertained that there was no antibody present in circulation. And what we found was that when these mice were challenged, there was no IgE, and they did not develop anaphylaxis. So it means that this antibody or this treatment was doing something else than preventing the formation of IgE. So in summary, what I've told you is that long-lived IgG1 memory B cells play a prominent role in the persistence of peanut allergy. Not IgE memory cells, which basically don't exist, or IgE producing plasma cells, which are not lifelong. Peanut exposures activate these cells, which then differentiate into IgE producing cells. Treatment of allergic mice and cells from peanut allergic persons with an anti-IL-4 receptor alpha antibody fully prevents the regeneration of IgE. And in mice, this treatment fully prevents anaphylaxis. These protective effects remain even after clearance of the antibody. And this may suggest that perhaps this treatment has facilitated some degree of immune reprogramming, so that the immune system does not respond in the same way to, in this case, peanut. And this is an area of very intense investigation in our lab. So if we go back to the beginning and to the question that Food Allergy Canada asked me to address, is it possible? And the answer is, it is not possible, but there's all kinds of reasons to believe that it will be possible shortly.
Great. Thank you so much, Dr. Jordana. That was a very informative presentation. I'm going to ask you to turn on your webcam at this point, and we're going to go through some questions. Now, we received a number of questions in advance from people that have submitted them during registration. And we also received a couple that came in during the presentation. So right now, I don't see, there you are, terrific. So thank you. That was a very great, insightful presentation on really getting into the science background. And I know there's many of in our audience that really appreciate that level of understanding. So the first question, I want to go back to what we talked about just in the introduction around the research findings, around whether they apply to all allergens and even ones that might be considered rare or idiopathic. What is your perspective on that?
Yes, I think that this is this will be universally applicable, and the reason is that, to the best of our knowledge in the scientific community, the basic mechanisms that I described today in terms of the regeneration of IgE are the same. Okay. So therefore, one even though we focus primarily on peanut, I have every reason to believe that it will be applicable to other food allergens.
Great. Okay. Now, what needs to happen next to translate these insights into a therapy, and kind of to this, the time frame? Everybody's interested in what's the time frame until it might be possible. So what steps have to happen, and what's your best guess on a time frame until preventing anaphylaxis can be possible?
So, Jennifer, let me say first that I don't think that in the field of food allergy, we've never been in the position that we are now, never before. And the reason is this: in the first place, basic research such as the one that I presented today, and other basic research by other people, they provide a very strong rationale for this treatment with this antibody to work. Very strong rationale. But now, in addition to that, this antibody already exists. And thirdly, this antibody has already proven efficacious in other allergic conditions, such as nasal polyps and atopic dermatitis. And to a certain extent as well, in severe asthma. This is very positive.
Right.
But that makes me believe that it's going to be even more efficacious in food allergy. And the reason is that in all of these other conditions, IgE plays a role. But there are a number of other things that also play a role. Whereas in the case of food allergy, it's exquisitely mediated by IgE. So
Okay.
if you have a treatment that is able to prevent the formation of IgE, I think that that is it's really amazing.
Right.
And
right.
the final reason is that this antibody is already on clinical trials for food allergy.
Okay.
So I think that there is the basic research provides the rationale. The antibody already exists. It has been proven efficacious in other allergic conditions, and there are clinical trials ongoing. How long this is going to take? Well, even these clinical trials have been slowed down because of the pandemic. It may be a matter of 2 years, maybe less, maybe a little bit more, but we are right there.
Right. No, that's very hopeful. And I'm assuming we're speaking about dupilumab. Is that the antibody
Correct.
that? Because that question has come up through our community around dupilumab and other biologics. And it does sound like there's biologics with these, there are clinical trials with these in conjunction with oral immunotherapy. Can you speak to us a little bit about how, because many in our audience may be familiar with OIT, but how might these work together?
Mm-hmm. Well, the two clinical trials ongoing, one of them is a monotherapy. So they look at the dupilumab only. And then the other is dupilumab along with oral immunotherapy. Now, I'm always been a little bit skeptical about monotherapy with dupilumab only. We'll see. The studies will tell us. But to me, the ideal scenario is the combination of OIT along with dupilumab. And the reason is that, I think that ideally what you want is to stimulate the system with peanut exposures, which is what OIT would do. But at the same time, we want the response, the immune response, to be deviated. So in a way, it's like, you step on the gas pedal and then you move forward straight. But you don't want that. You want to change directions. So, if you step on the pedal and then turn the wheel to the left or to the right, then you go into a different direction,
Okay.
which is exactly what you want. And
Okay.
ideally, under these conditions, maybe you can reprogram the immune system. Now, remember that, there's a lot of things that still we don't know. And so that's why, that's why in the lab, we're so very busy trying to understand whether or not this treatment can induce reprogramming.
Okay.
Why is this important? Well, because if it doesn't induce reprogramming, it means that this treatment would have to be taken on a regular basis forever. However, if it induces reprogramming, it would suggest that at some point in time, you can stop the treatment.
Okay. Okay, we are running a little bit short of time, and I want to close with one question. Okay, because it's, everything seems to, a lot of our conversations circulate around COVID. Okay, but I would like, the question that has been asked around, are there any learnings from whether it's the mRNA science or the fast tracking in the development of the COVID-19 vaccines and treatment that you think can be replicated for food allergy? And then we'll have, okay.
No, and I would, I would love that things were differently, because I'm a food allergy researcher. But the reason why the COVID vaccines have been fast tracked so vigorously is for two reasons. The first one is that governments have pumped not millions but billions of dollars into the pharmaceutical companies to develop those vaccines. But also there is another element, and the other element is that, well, as you've seen, people with COVID infections, it's a pandemic, and then we've had huge mortality. Thank God that's not what happens in the context of food allergy. So I think that that society and governing bodies would see a very different sense of urgency in one case versus another.
Okay.
Sadly,
well, listen,
sadly
yes.
for us.
Yeah, but we do have, what you've presented here today provides much optimism and hope. And I actually would like to kind of close off this portion of the webinar with a comment that we received on the chat box, because I think it really summarizes what we're hearing from people. And the comment is, I really appreciated everything about this presentation, especially the scientific detail and the promise of what Dr. Jordana and Dr. Wasserman's lab is studying and has found. Please pass on my heartfelt thanks to Dr. Jordana and his work, and thank you to Food Allergy Canada. Thank you for putting this webinar together. So we really appreciate that comment. And thank you, Dr. Jordana, for your time. I'd like to make a little bit of comments about Food Allergy Awareness Month and about a survey that we would like everybody to complete. So just as a reminder, you'll get a short survey through GoToWebinar immediately after this, and we would ask you to complete this. You'll also get one in your email in the next hour, so take a moment. We really want your feedback because it helps us improve our future webinars. In terms of Food Allergy Awareness Month, that's this month, we have launched a Know It, Treat It campaign. And I'd also like to mention a couple of webinars that we have scheduled this month. We've added a new one on May 11th, next Tuesday, on COVID-19 vaccines and food allergy. And is any approved vaccine the best vaccine is the question we're going to explore with experts from Health Canada, from infectious disease, as well as from the allergist community. We also have another one on May 19th, epinephrine, first in the role of Benadryl in anaphylaxis with Dr. Wade Watson. So you can sign up for both of those webinars on our events page. As those of you who are in our audience are aware, many of you are aware that Food Allergy Canada is a not-for-profit charity. We're completely reliant on your donations and would greatly appreciate it. If you could consider a donation, you can visit foodallergycanada.ca/donate to learn more about the impact your donations make. And we would also like to thank our sponsors for our ongoing webinar series, the Walter and Maria Schroeder Foundation, the Sean Delaney Memorial Golf Classic, EpiPen, and the Peanut Bureau of Canada. Thank you for your participation in today's webinar. You can view a recording of this webinar at foodallergycanada.ca shortly, and please share this with others who you think may benefit from this content. This now concludes our webinar.